This talk is a compromise. The first version of the title was just this. Okay, and I thought that that would then be a challenge, and then probably have everybody leave right after lunch. Anyway, I have a lot of thank yous. I mean, I'm very grateful to the Simons Foundation. I've been associated with the things that they do for so many years, and I've heard so much interesting and fun stuff here, and it has a really unique perspective on how to support mathematics and science. Anyway, so now I want to do the thank yous first, just in case it's the only comprehensible part of the talk. Okay. So firstly, I want to thank Rich and Tuka that dragged me into NITMB, and then I have collaborators. With Rich and Sam and Lorenzo and Ryan and Evan, we've been talking about flywings, and Rich discussed those at an earlier meeting here, so I won't really be discussing them. So by the way, I feel like Mathematicians Anonymous. I'm Shmuel Weinberger, I'm a mathematician. Okay, and worse, I'm a pure mathematician. So, I mean, we looked at lots and lots of data, and then somehow at the end of the analysis, we saw some kind of low-dimensional structure was governing what potentially was an infinite dimensional space of flywings. Okay? You just go ahead and you look at these things, and I mean, it sort of locally looks two-dimensional at most points. There are some places where it's hard to distinguish from being three-dimensional, but it might be that things are crisscrossing. There seems to be some very interesting structure there. And I tried to understand how in the world that could be from an evolutionary point of view. And I'm not interested in the correct answer, okay, because I'm a pure mathematician. I'm interested in how could it conceivably be, okay? So I have a very, very primitive, false point of view on evolution, where I just think that, it all gets to be better and better and better, right? Survival of the fittest, which means that things should then be concentrating on a point, right? Which means that there shouldn't be diversity at all, okay? Which is, of course, the point of view of the current administration. But I wanted to understand why is there diversity and why is there, at all, rather than just being, the optimum. Okay, so you could imagine, okay, well, I'll get there in a second. And then why is it so small, right? There is some, but then it's, why could it potentially been infinite dimensional? Why is it only, 2 or 3 dimensional generically? Okay, and so that was what was, the perplexing thing to me that I was observing. And I'll talk about this could be a cautionary tale. What happens when you ask a pure mathematician to just think, okay, unmoored from things. So anyway, I'm not going to talk at all about flywings or anything like that, but flywings were part of it. And also a very intriguing suggestion that Tuka made to me when we were bullshitting about evolution and, and gradient flows and, convergence, and you go to critical points. And, there are lots of people who have observed that, when you do a gradient flow, the critical points of higher index, you're supposed to be converging to optima. And everyone worries about the fact that there are many, many optima, right? Many, many local minima, okay? Yes. So, I'm using that convention. Okay, so evolution moves you downwards, not climbing Mount something or other. Okay, so of course there are many of them, and you're worried about the fact that you might be converging to the wrong one. But the speed of convergence is associated with these higher index critical points. Okay, that has a very, very big impact. And the thing is that we have, unfortunately, only finite lifetimes. Very finite lifetimes. And it could be that the things that we're observing when we see things macroscopically, might actually be associated not to local minima, right? I mean, it could be associated to critical points of higher index. So part of the ideas that are percolating have to do with not only measuring local minima, but understanding phenomena associated to these critical points of higher index. So that's sort of another thing to keep in mind, or take home, or something. Okay, so this talk is not a contribution to biology, so hopefully it should be a contribution to mathematics. And there will be some new theorem here. And in fact, this is an example of biomimicry, okay? So the first meeting I went to with biologists was actually a meeting at the National Academy of Sciences, and it was biologists and roboticists, and it was trying to go ahead and use ideas from biology and the observations in order to design robots. Of course, machine learning is another great example of biomimicry. You know, many of the motivational ideas come from looking at various biological templates that seem to work according to our understanding. Now, of course, in the end, when you use it, you might not be reproducing the biology, but if, you know, the proof of the pudding is in the eating. Okay, so I'm going to describe a theorem that in the end was motivated by my poor understanding about the way enzymes act, okay? But hopefully I'll get to that point. So my collaborators are Jonathan Block and Fedya Manin, and if you want last names, they're on the slide. Okay, yeah, so, okay, so by the way, I'm in love with analogy, okay? And, you know, I mean, when you want to be scientific, you don't call it analogy, you call it abstraction. Okay, but it's sort of the same thing. And one of the things that, you know, that the way I would think about biology, I saw in a book by Gaddis on grand strategy, which is, I guess, the things that generals are supposed to be doing. You know, the problem that you have the theorists, and then if the theorists want to be correct, they tell you things that are pretty much useless. And then you have the historians who tell you about all of the exact detail. And, you know, and it never applies to anything else because it never repeats. And it just struck me as being, you know, very apropos of a lot of the discussions and frustrations that I had in trying to think about this. So I want to talk about, so the compromise version of the title, okay, which had English words in it and geometric metaphors in biology, so I want to, you know, the fact that the same metaphor, the mathematicians like studying the metaphors. Okay, that should be the thing. And then if a metaphor has many, many instantiations, you could then ask, you know, which of the, you know, what are the parallels among all of these things? Okay, so, you know, there are lots and lots and lots of networks that come up. Okay, and then, you know, at the very, very end, I threw in one that's, you know, threw in a bunch that are not biological. Okay, and, okay, so these are some set of things that you could try to study. Okay, this is an old one, right? The tree of life. Okay. Okay, and then there's another metaphor that people like talking about a lot: landscapes. Okay, and they're also everywhere. And you know, so okay. So the question will be, you know, if you know, for you know, given that my goal is to prove theorems, right? I mean, things that are suggested in any one of these settings that make sense, you know, are grist for the mill. But one should wonder about the fact that the same metaphors are being used over and over again. What are the connections among these? I saw this picture in I don't know how many talks since I got involved in the NITMB. So, you know, I love that picture. Okay, here's another geometrical picture. Okay, I pulled that one off Reddit. This is called Galton's polyhedron. So Galton is the you know, Galton-Watson trees, sort of well-known probabilist, eugenicist, cousin of Darwin, I believe. This was from his book. I mean, because he couldn't draw 3D pictures. Okay, back then life was two-dimensional. Okay, I don't believe that that's a picture of Galton, but it, you know, it was good enough for Reddit. Yeah, so he sort of thought that the faces of this polyhedron were species, okay, and that he was wondering really about evolution. He thought that these kind of things, you know, were the stable parts, and that some force or other would move you from one one stable version to another. You know, like imagine you put a dice on top of a vibrating drum, and you know, typically it just stays where it is. Every now and then it gets energetic enough to push to another one. And you know, he spent some focus on the fact that this thing here is skinnier than that one and stuff like that in his model of evolution. So anyway, the moral about this is that everyone wants to be a geometer, okay, right? You're all studying geometry. And so anyway, so what I want to do is, firstly, what do I want to do? Okay, I'm going to be showing you some mathematical techniques and ideas. Hopefully they'll be useful, because I think that these metaphors are occurring frequently. So even if what I'm saying is, you know, some kind of science fiction, right? Hopefully, you know, some pieces of it, right? Compelling fiction, you know, has various paragraphs in it that feel like they could be true. Okay, so if the whole story doesn't make any sense, so be it. For the mathematicians, there's actually a new theorem. And this idea of biomimicry, I think, is really valuable to pure mathematicians. There are some very, very subtle, very unexpected phenomena that occur in biology, and that people should, you know, just going and observing and trying to absorb what these kind of things, you know, how these things can happen, you know, is really useful. Okay, and then whether it's going to be relevant, that's a hope. Okay, but you know, maybe a matter of faith that it will be. But okay, so I'm going to tell you the theorem. Okay, so that there's something, some substance, and then I will have to explain what the theorem means, why we care about it, and then I'll explain to you where the enzymes come in. Okay. Okay, so X and Z are finite complexes. Those are kinds of spaces. Okay, so if you think you know what a space is, fine. Simply connected, okay, I'll have to explain that. Then you're going to look at the functions that go from X to Z. Okay, so you might have an example in mind. Okay, say I'm mapping the circle onto the two-sphere, right? So I could have lots and lots of functions. Right? How would I do it? I might imagine 3 real valued functions, such that the sum of the squares equals 1, whatever it is. However I want to think about functions from the circle to this two-sphere. That's a big infinite dimensional space. Function spaces are infinite dimensional. Okay? And then persistent homology, I'll have to explain what it is, although it was tacit at a moment in Rosemary's talk. Okay? It's saying something about a calculation of the persistent homology in terms of the ordinary homology, okay? Up to bars of some bounded, okay? Blah, 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 because it's an abstract statement which I don't expect you to know or understand, okay? But by the end of the talk, I hope you'll appreciate the kind of things that it's saying, okay? Okay, so that's what it is. What do the words mean? What's the context for it? Okay, it turns out that this theorem answers some questions that are about 50 years old, so I'm kind of proud of it. And towards the end, hopefully, I will make some remarks about why I hope that biologists will pay attention to it. Okay, here's another picture. Okay, so this is you know, a famous pontylist picture. Okay, and of course, the point of pontylism, never said that before, is that you just have dots, right? I mean, right? You don't have the continuous object, okay? Right? But that, you know, when you look at it from the distance, you actually go ahead and you persert, you know, right, all of these points, right? You know, if you know, people who know about topology might think a finite set of points is a finite set of points. There's nothing else to it, okay? But nevertheless, we go ahead and we see a picture, right, out of this. And this has something to do with the emergence of continuous structure from the discrete. Okay, you could look at it the reverse way, that you study continuous things from discrete samples. But I want to adopt the opposite point of view that the continuous emerges from the discrete. Okay, so now I'm going to move to the blackboard pretty much for the rest of the talk. Okay, so that's a space, okay? That's a space. A topologist is someone who doesn't know the difference between this space and that space, okay? All right, so far so good, right? I mean, I'm hoping this is a dialogue, not a monologue, okay? So but one of the really good things about the modern point of view, right, is that the spaces that you can think about don't have to be embedded in the blackboard. They don't have to be embedded in 3 space. Okay? They don't have to be embedded in any Euclidean space at all. Okay? So there are abstract spaces. Right? I might start with the sphere and consider pairs of antipodal points. Right? So that a single point in my new space is a pair of points in the old space. Okay? So people who know about this will say, oh, that's the projective plane. Okay? If you try to realize it, you can't fit it into 3-dimensional space. If you wanted to realize it concretely, you need 4 dimensions. Okay? That's not my interest. Okay? The thing that's of value is that you should be able to think about abstract spaces. Okay? Right? Another examples are configuration spaces, phase spaces, tangent spaces. I mean, all of these are abstract spaces that you create if you, for example, have something involving the real numbers and you're looking at functions on it that are periodic, right? That leads you to consider a quotient space of where you identify points with their x is identified with x plus 1. When that 1-dimensional object you then frequently will draw in the plane, right? But of course, when you parameterize things, you'll still use one variable, theta. Okay? So this thing here is called the 1 sphere. Okay? It's 1-dimensional. The topologist doesn't look at where you happen to embed it, because that's just a crutch, right? For how you're going to maybe visualize the space. Okay? And a lot of what I want to do is an idea that Thurston taught us topologists, which is to not look at the space from the outside, but to look at the space from the inside. What's it like to live in a space? Okay, so okay, so those are abstract spaces. So let me talk about another one. So if I have two things, they might be species, they might be proteins, right? And I could then consider combinations of them. Okay, so this means, I'm not going to care about the whole amount. I'm just going to care about proportions. So this would be all A, no B. This would be all B, no A. Okay, and then I have this thing here, which is measuring how the relative proportions. Okay, so thinking about any 2 species in this way leads me to the thing that we think of as being the interval. Okay, nothing great about that. Okay, but now suppose that there's a third thing, a third species. Okay, so then so this is the beginning of like networks, right? You begin looking at the things and you ask about their interactions, and that kind of stuff. We tend to draw these graphical pictures. But I'd like to emphasize, that there's sort of a different thing you could ask. Is it the case that there's, say, only enough resources to support 2 species, but there's never enough to support 3? Okay? So when you're dealing with a situation like that, you're really dealing with, right, the 3 intervals, okay? Except that the intervals are glued together along the special situations where the ecology only has 1 species in it. Okay, the topologist, of course, will say that's the same thing as a circle. Okay, who cares? All right, but this is a very general kind of construction, right? You could imagine, and I will hope you imagine, right, that these things, say, are proteins or a very, very large ecology. Okay, and then you might have this connected to a D, and here I might have E, F, G, okay, and some combinations of all 3 are present and others are not. And all of a sudden, these kind of configuration spaces, so these kind of things are sometimes called simplicial complexes or hypergraphs, depends on your tradition, I guess. Okay, but if you have, say, 4 species, okay, and any combinations of 3 are possible, but not all 4, then you're having what will be the boundary of a tetrahedron, which to a topologist is a 2-dimensional sphere. Okay, and if I have n species, the boundary will be an n-1 dimensional sphere. And now, if I'm thinking about gene expression, and it's going to be expressed in terms of proteins, okay, I'll have many, many possible proteins. And I'm only going to imagine what's being produced by a small snippet, okay? So it's not going to be all possible proteins, okay? Because it turns out the space of all possible proteins, whatever that is, it's some very high dimensional simplex if we're allowing arbitrary combinations. But if we're imagining what's produced by a small snippet of DNA, I'll have some simplicial complex. Okay? Which will be, depending on what this length of the snippet is and the amount of things that would be, which simplicial complex you get will be different. That's an empirical question about which simplicial complex, should be modeling this. Okay, and so all of these things, all these crazy spaces that topologists look at, potentially they all come up. Okay, and to a topologist, there is this thing called triangulation. If I start off with a sphere, okay, this is the way people do things on a sphere in a computer, is they put in meshes, and all of a sudden they turn weird curvy objects also into this kind of simplicial picture. So, sort of the whole panoply of geometric mathematics and topology is in principle, could have been related. It's not necessarily unrelated. Okay? Okay, yeah. Yes.
Get a little bit more grounded. I remember, so when you had the pointillism, you had the Seurat, right? Mm-hmm. You said that we see the objects and shapes. Now I could imagine that if we didn't have the prior, some locality prior, some physical, and we have a notion. Our visual systems have evolved to do the job of processing natural scenes for us, and to make. So we have priors about the continuous form of objects, and that objects don't teleport. Objects have physical extent. Mm-hmm. So when you say that you want to look for the discrete spaces that become continuous, are you thinking about imposing that kind of prior so that those correlations turn into objects, those higher order correlations that we see between the points turn into objects? Or are you going to be agnostic about that when you make this metaphor?
So I was originally planning for that to lurk behind the scenes, okay? Okay, so no, but the thing that you should have in mind is that, so if you just have the topologist's version of a discrete set of points, then it's just as dust. There's no relationship among those. Okay, so you have to so you have to beef it up with a certain amount of geometry. So you should imagine that you have points together with some distance between them. Okay, and now the thing that I ask you is, so here's the thing: if you imagine the lattice constructed in dimension, from the plane, okay, firstly you would never ever get confused with the integers sampled from the real line, okay? And there are a lot of reasons why you wouldn't get confused, okay? One reason might be, well, if I look at how many points there are of distance at most r, in the line it's about r, and in the plane it's around r squared, okay? But I hate to do this to you, but if you did the same exercise from the hyperbolic plane and hyperbolic 3 space, okay, in both of those, the number of points in a ball of radius r grows exponentially in r. You can't tell dimension apart from that perspective. Okay? So the question is, what would you do? How do you infer hyperbolic space from a lattice inside of hyperbolic space? Okay? So Rosemary already told us the answer. Okay? Which is you begin drawing balls around each of these points, and you okay? Fictitious balls because the other points don't exist. So in other words, you measure at what time, I take these points and they're distance 1 apart. So at distance 1, I have some kind of lattice of points connecting to each other, a big graph. But I have that both in the hyperbolic plane and in hyperbolic 3 space. I go to a further scale, and in the plane, what happens is more and more fills in, and it begins looking planar, okay? And then in 3 space, it still looks now like a web in 3 space, okay, right? Now what happens when you go to yet a further scale? For the plane, what happens is it doesn't grow any bigger, okay? I mean, there are essentially no features that grow beyond a certain scale other than the 2-dimensional size. It turns out all the 2-dimensional aspects of that web die by the time you get to a larger scale when you sample from hyperbolic 3 space. Okay? Okay, right? So that's an example of you're able to detect a large scale dimensionality, right, rather than small scale dimensionality from from discrete measurements, and here it was scale, by going to larger and larger scales, right? Which sort of feels weird for the inferring continuous from discrete, but nevertheless, it's what we've learned is sometimes the right thing to do. Okay? Yeah. Thank you for that question. Okay. Are there any other questions? Okay. So let me point out, okay, now I'm not going to tell you what simply connected means, but I'm going to tell you in this context something that would suffice for it being simply connected. And I think it's reasonable if you're going to be imagining gene expression as being your source of simplicial complexes, okay, which is that whenever I see A, B, and C, I actually can find all 3 of them together. Right? I'm looking at snippets, and if it could, if it could produce A and B and C, then I could produce, I'm at a scale where I don't have any kind of empty holes. Okay? Okay, that's an empirical question again, whether it's really true, but it's my guess. Okay? A hole like this, and if it's not true, that would be a valuable thing to know. Okay, that some combinations of 3 proteins don't occur in snippets of this size. Okay? So now, if you have something like this, and you're in this space X, and so here's X, that's a picture of X. Now, if I imagine any closed curve in X, what does it look like? Okay, well, I mean, I'll approximate it by, this curve is going through this triangulation of the space. So I'll just move the curves, move a point here to a vertex. So x0 is here, then this is x1, this is x2. It'll then go to a point x3, and so on, okay? Okay, and then, right? So I'll just be able to shadow the curve by a sequence of vertices where each vertex is touching the next. Okay, now, so this was simply connected, and now the reason that this connects to what other people might call simply connected is the following. If I have a curve like this, okay, so there's no hole here, so I could deform it to a shorter curve. Right, just imagine pushing it. Okay, so for people who remember their complex variables, if you have domains in the plane, if it has a hole in it, then it's non-simply connected and some theorems don't hold. Okay, right? So that was the condition, the sort of no holes condition, and it means that if I have any curve, there's this very greedy method. There's a greedy algorithm to shorten any curve, right, up to deformation. Okay, so to be simply connected means you could shrink any curve up to deformation, but actually these are, have very special geometry, okay, if they're simply connected for this reason, right, which is that a greedy algorithm actually shortens them. So, so another simply connected space that you're used to is the plane, and if I have a curve of length r, like this circle of length r, so it has, I guess it has length 2 pi r. Okay, we're friends, who cares about that? Okay, right, it tends to bound something of area r squared, right? When I shrink it down to nothing, okay, this is the behavior that you see. Here, if I have something of length L, right? I just used 1 unit of area to shrink it down to L minus 1. So here, curves of length L, in this geometry, I want you to feel like you're in the space, okay? Right, so you shouldn't feel like you're in Euclidean space, right? Where curves of length L that, it's simply connected as well, but curves of length L take like L squared to fill in, right? In gene expression space, presumably, curves of length L can be filled in, but they only take area L to fill in, okay? Okay, and right, now there's one other kind of space which you need to know about for understanding the theorem, and that was, just the maps from X to Z. And I already told you about that. I'm looking at the continuous functions on one space, taking values in another space. Okay? If you could think of it, you could make a space of it. Okay? And that will be a big, complicated space. If X were just a point, this function space is easy. It's just another name for Z itself. Okay? If it were 2 points, then it would be Z cross Z. Right? So, but once you have infinitely many points, this becomes infinite dimensional immediately. Okay? So there's a question about how to think about it. Okay. All right. So what? Okay. So by the way, people tell me that if you're a physicist, all functions are automatically differentiable, et cetera, right? I mean that, that only mathematicians worry about certain pathologies. So again, Lipschitz functions are the same thing as functions if you're a normal human being. But not Tuca, actually, right? Because Brownian motion is Holder and not Lipschitz, okay? But, all right. I apologize. Okay, so a Lipschitz function. So remember, I said that all of my spaces will have metrics, and I don't. So to say that F is Lipschitz means that if I look at the distance between what F does to various points, there's some number L such that the distance in the target is at most L times the distance in the domain. Okay? Now, there are theorems that the space of Lipschitz functions is dense and it's homotopy equivalent to the space of all functions, but who cares? Okay? What's the meaning of this? Right? So F might be something that you're trying to observe, right? You're having X being something, right? Some latent variable or something, and you're trying to observe something about it. And so continuity means that if I measure enough decimal places, in the domain, I'll be able to get some information about how close I am in the target. That's continuity, okay? And Lipschitz says the number of decimal points you need in the domain is equal to the number of decimal places you need in the range, up to a finite amount of error. So if you multiply by 100, okay, you have a Lipschitz constant of 100, you lost 2 decimal places. But, the extra need, you don't need that much more information, okay? Now for Brownian motion, you need double as many decimal places as in time for the value of the function. So that's sort of a weird thing, okay? All right, so L is called the Lipschitz constant, okay? And I could think of it as being the it's the sup of d of fx, fy over the distance between x and y. Okay? And I want to think about this as a measure of the complexity of a function. Okay? Because if I know a Lipschitz constant, then I could begin discretize. I could know at what scale I have to study things in order, to do a measurement. So if I'm trying to do a measurement at size 1 in the target up to 1 inch, so then in the domain I should be sampling at a rate of 1 over L. Okay, and that way I will be able to get so if you want to put a function into a computer, the easiest functions are the Lipschitz functions. Okay, and now you can begin studying spaces of Lipschitz maps, and I'm going to be interested in this as being a functional on the space of maps from X to Z. Okay, so I'm having the maps from X to Z, and I'm going to be studying log of the Lipschitz constant. That's a technical issue. Typically, it's because I like turning multiplicative errors into additive errors. Okay, so that's what the log is there for. Okay, it fits in better with the conventions of the literature on persistent homology. Okay, but anyway, so I'm interested. So now, if my Lipschitz constant is 0, okay, then my function is a constant. Okay, so it starts off and the constant functions, I mean, aren't boring. They're the space Z itself, and if I love Z, okay, happy days. Okay, but now, yes.
I just have a simple clarification question. So you told me that if I'm a physicist, I can think about this as any function.
Yes.
And I started to think about the exponential.
Okay.
And if I consider intervals of 1, that gap can get arbitrarily large in terms of the exponential.
Right. Okay.
So
Yes.
Are we like bounding the space or something like that? Or?
So I'm going to be mainly interested in when X is a finite complex, and so is Z. Okay? If it's infinite, then if they're infinite complexes, then you have to do something else. And putting the Lipschitz constant on for a non-compact domain is actually an act of violence. Okay? And the tradition is to say you're locally Lipschitz or something like that.
Okay.
Yeah. Okay. Yeah. Yeah. So think about the situation where the domains are bounded. Okay, as we're thinking about this. Okay, so again, if I ever get back to DNA, okay, the domain will be a circle. Okay, now why is it a circle? Well, you have a strand of DNA, but it always starts off not doing anything at the beginning. So it starts off at the empty protein. It ends off at the empty protein. Okay, so it's a closed curve that starts and ends at the same point. Okay. The reason there are good mathematical reasons to also prefer, if I wouldn't close it up, okay, then there's nothing that would prevent it from shrinking the trivial way. An interval of length 1 is more or less to a topology, you shrink it to an interval of size 0.99 just by speeding up what you're doing, or, or stretching it out, right? I mean, there's some kind of easy deformations. Okay, once you begin putting the circle in, it prevents there from being easy deformations. Okay, so ultimately I'm going to be making the suggestion of a possible place of relevance to biology would be to look at the space of maps from the circle into local gene expression space, right? So the idea would be you have this long thing, you look at each point in your discretization, you look around it at some scale that needs to be discovered, what's a useful scale. Okay, and then, as I move along my curve, I'm having this moving snapshot of what the expression is, and that describes some path that goes right. Global gene expression should be viewed as a closed curve in gene expression. Okay, to push the biological metaphor. Okay, so anyway, so let's think about this. Let me actually do a case. I'll map the circle to the circle. Okay, and so we started off where the Lipschitz constant is 0. We get the constant map, so that's just the circle. Okay, now if the Lipschitz constant gets a little bit bigger, nothing much happens. Okay, Okay, so here's the target circle, and then I'll draw the domain circle wrapping around it. So it starts off being at a point, but then when the Lipschitz constant gets a little larger, it wiggles a drop. Big deal. Of course, if you're an analyst, you could study this. This would be a function from the circle to the real line, right? You could use the angular coordinate, and okay, so that's analysis. Okay, fine. Okay, now, once the Lipschitz constant gets to be the magic number 1, though, you have these kind of deformations of the smaller ones, but all of a sudden there's a brand new function, namely the identity map, okay, which goes all the way around. Okay, and now this function, okay, if I try to come up with an angle to define this function, it gets a discontinuity, right? So we all know that you have to go ahead and, right, angles are between minus pi and pi, right? There's something or other you do, and I just take a point and I try to pick a real defined angle, and when I come back, I could measure how many times did I go around. Okay, so in order to go around n times, so if I'm going to view this as the unit complex numbers, I would go z goes to z to the n, and that has Lipschitz constant equal to n, and you can't shrink that any smaller. Okay, so if you begin looking at this function space, right, from the point of view of increasing complexity, okay, right, there's the bottom complexity where things just look like constant functions, goes for a while. The space is not connected, so this is the beginning of nonlinearity. Okay, that I was able to think about this as being a linear thing. As long as the winding number is 0, then you could define a consistent angle. Okay, and then you can study it that way. Okay, so that's perfectly good. Okay, but these other ones you can't study the same way, and it's sort of a nonlinear kind of phenomenon. And a very basic fact that that I won't explain, is that, in fact, that's sort of what happens. If I have any Lipschitz function, and I go ahead and I now imagine trying to do something like gradient flow on the space of Lipschitz functions, it will keep going down until it hits z goes to z to the n, where n is positive or negative, could be, okay? And that will be all that happens. Okay? Fine? Okay? All right. So components of the space of maps from x to z, these are sometimes called homotopy classes of functions. Okay? So this has been an object that algebraic topologists have cared about since the beginning of since before the beginning of algebraic topology. Okay, this fact about the circle is sort of the first non-trivial theorem in the subject. Okay, and and they really care about it. I mean, when x is the 254-dimensional sphere and z is the 128-dimensional sphere, there was an unknown question that was solved last year with a and the archive preprint got modified last month. I mean, this kind of stuff is hot. Okay, okay. So anyway, so the question is, what happens to this? Okay, so. Okay, I'll return to that. So this subject has really existed, and there was some prehistory where you which looked like the case of this circle, where there's this nice geometric way, right? It's very easy to understand winding numbers, right? I could think about it in terms of the defect in the angle. I could take a point, and I could look at the pre-images and count them, keeping track of whether it's going in the same direction or the reverse direction. Right? There are sort of all of these things, and so we understand, right, that was a very, very concrete, easy picture for what happens for the circle. And the basic motivating question was, in the 1950s, Jean-Pierre Serre introduced algebraic topology, introduced algebraic methods into the subject called spectral sequences. And all of a sudden, it made this kind of thing very calculable, right? What are all the components, or even the higher dimensional features of these spaces? Okay, and then in the 70s, Gromov asked, do we really understand anything about this? You go to the oracle, someone who's well-trained in the dark arts, and they go ahead and they do these calculation after calculations, right? And you want to know, what's the result of it, right? So you're having this function space, and you want to navigate it, right? You're told you know the components, so so there's maybe some algorithm that you could compute, right? That tells you I could connect this point in the space to that point, which means I could connect this function to that function. I could deform this map to that map. Okay, right? That's what the upshot is, right? So, so there's a theorem that the maps from the 5-sphere to the 4-sphere, that there exist exactly 2 maps up to deformation. So, and can I tell? Yes, there are algorithms to tell. But now you ask, okay, suppose I have a concrete one. I have a map of one complexity. How hard is it going to be to move it into the normal form? Or, there might be some specific maps that have that I care about. I might care about harmonic maps. I might care about maps that satisfy some differential equation, right? Solving these things involve moving along this terrain, okay? So basically, the work that I'm trying to explain, not very successfully, is about that problem. Okay? So now I'll tell you what the theorem says. okay? What the theorem says is that if the x is simply connected, okay, and it has some other special property, okay, which, then if I have two things with complexity L that can be deformed into one another, so there I ask the dark arts people, okay, then when you have a special property of it, then you could actually deform one to the other without increasing the complexity more than linearly. Okay? Now, linearly is sort of a reasonable amount because that's independent of which metric you choose on the target Z. Okay, if I have a let me just, this is sort of okay, this is the usual two-sphere, and here it's very easy, if I take any simple closed curve, I can deform it, to a great circle or something, okay? And then I take the great circle and I could push it further down, right? I mean, there's the thing you do by gradient flow, and you might get stuck on sort of great circles and things like that, and then you push it down, so we understand what happens. But now suppose that I'm a topologist and I grow a couple of mushrooms here, okay, which you could easily imagine, okay? And now, if I have a closed curve like this, okay, it doesn't, it needs a global move, okay? I mean, I can't just do gradient flow. It gets caught on this. And in fact, the number of bad curves of length L grows exponentially in L because there are two mushrooms, and it could do them, any word in A and B I could mimic, okay? So you see that the number of places to get stuck grows exponentially, okay? And in fact, the generic situation is if I have a d-dimensional domain, the number of places to get stuck grows exponentially in L to the d. Okay, so it's super exponentially if I was looking, instead of maps of the circle, I'm looking at higher dimensional maps going in, right? So it's, I said you put them into a computer, but I was lying, right? It's super exponential, okay? But still, the theorem says that for suitable things like spheres or homogeneous spaces and things like that, it is a case that you never have to increase complexity more than linearly, although the final clause of the theorem is that it's not true in general. Okay, so that had been so these questions about increasing complexity were asked by Gromov in the 1970s, and we solved them, in some cases before, but some cases this year. And one special case where the answer is always yes is when the domain is a circle, and we were motivated to actually ask that question because sort of all of the literature on these kind of things, it turned out it was the geometry of the target that was relevant. So for example, the situation that we saw on the circle, that happens whenever the target is a non-positively curved space. So it holds for tori, it holds on the Klein bottle, okay? On hyperbolic manifolds, right? That kind of property where, in the natural metric, you do gradient flow, and it just goes down to the most beautiful map. And then any metric on those spaces, add on as many warts as you want to a torus, it's still going to have, it won't now be by gradient flow, okay? But the critical points of index 1 that it has to go over, now we're going back to Tukka, okay? Those are only linear in size in the length of the curves, okay? Now, of course, it doesn't apply to the torus, it's not simply connected, but so for homogeneous spaces that are targets, that's what the theorem says. So by the way, so where are the enzymes? Because I really should be wrapping up rather than getting to my third index card. You see them right over here, right? Imagine that you had a thing called mushroom ACE, right? And if I had a mushroom ACE, that's what gives me a little bit of, something goes ahead and snips the curve, inserts some extra rope over here, okay? And now that rope is enough to go over this hill. And then that thing goes off, okay? And then, you come back to the next one, you use mushroom ACE again, okay? You do it and right? I mean, perfect catalysis or something, okay? And that will go ahead and work on the sphere. So that isn't enough to prove the theorem, okay? This is an idea that you want to embed in the proof, but it applies to a certain class of spaces, okay? Namely spaces with finite homotopy groups. And then for the other spaces, you then do analysis. Okay? That you sort of say that this is what, that there's something or other that you do using differential forms. So like here, the relevant differential form would be the thing that you call d theta. The angle doesn't make sense, but d of the angle makes sense. Okay, so on the space Z, you have to look at the relevant differential forms. And that gives you some kind of analytic guidance, but there's, just no guarantee that you could geometrically mimic what you see analytically, so that's where the enzymes come in. Okay, so anyway, so that explains what the theorem was about. Okay, are there any questions? Okay, snowed you all, okay. Okay, yeah, okay, so yeah, I think I am not going to. Okay, so let me just make some additional comments about so this talk was hopefully entertaining. It's, you're after lunch right before you leave. I hope that it succeeded at that. Right, I introduced you to why thinking about high-dimensional objects is a good idea. I hope I showed you that rather than thinking about genetics as the Boolean cube, okay, thinking about it as a function space, you see the Boolean cube has this exponential growth in it, right? I mean, that finite dimensional objects don't have, right? So even though it's a discrete object, finite dimensional geometric objects, the things that sit in Euclidean space in a not too distorted way, they just don't have that kind of exponential growth, but function spaces do, okay? And thinking about it from the point of view of maps from the circle in, keeps into account some of the geometry about nearby genes that gets lost if you just think about it as a function from the Boolean cube. Okay, so those are some of the things that we've done. The idea of understanding the heights of these critical points, for people who think about statistical mechanics, right, you try to go to the ground state, okay, and this is, you do the gradient flow and you end up at a local minimum, right, and then you get stuck there, right, but then you begin having some thermal action going on. So now the stable state just depends on the heights, the values of the functions at these local minima. However, if you think about transitions from one state to the other, those are rare events, and whose time depends on something. And what it depends on is how long does it take to connect this local minimum to that one. It tends to always follow the shortest route. Okay, almost always when you see this transition. So it's exactly the kind of thing that we were studying, right? When do these components connect to each other? Okay, so studying pH1, studying the things that are absent from topology, right? Topology cares about the holes. It cares about are you connected or not. But here you're going to be measuring something more quantitative, which is how high does the function have to get in order to connect one thing to the other? Okay. There's another aspect, which is that these things tend to be, in some cases, small world networks. That actually the shortest path connecting one thing to the other in a component is much shorter than you would expect, given the fact that their volumes are growing super exponentially fast. Okay, so that's sort of another feature that comes out of this. Anyway, so looking at log L, that has the frame of making it topological somehow. Okay, but there's sort of a certain amount of, you lose the information, right? You lose a lot of geometric information, which might be interesting. When you might care about, does your sphere have mushrooms growing on it or not, right? So then log L throws away data of that information of that size. And I'm not, I've never studied it at a finer scale than that. There was never a topological reason to. But if these models make any sense, and even if they don't make sense, it still might be interesting to try to figure out what kind of geometry comes in. And are there other functions which are maybe more motivated biologically that one could try to study? I talked about the transitions being like thermal, right? That there's some kind of temperature. So that's some amount about how mutable this kind of thing is, and that shouldn't be constant everywhere on the space. Okay, so a part of the talk I didn't give is about random walks on these spaces that are where the randomness is modified by gradient flow, stochastic gradient flow on these spaces. And what turns out to happen is if the domain is more than one dimensional, you always drift off to infinity. Okay, rather than staying in critical points, and that the ground state is, which might be why if we had two-dimensional DNA, evolution wouldn't take place because everything just sort of pops out and moves off to infinity. There might be some reason that we encode things in one-dimensional structures, or if we had two-dimensional DNA, it probably would not be arbitrary continuous functions, right? There might be some kind of local differential equation that says that you can't, which would correspond to some kind of error correction going on. But anyway, so studying random walks where the temperature is non-constant seems interesting and important. Let's see. Yeah, so changing the fitness. Also, all of this was imagining a single function, right, rather than a set of functions, right? Evolution really takes place on populations, not individuals. This was, just an individual kind of thing. What is X? Is it simply connected? Okay, we translate, okay, etc. No sex, I'm sorry. But anyway, right, right, all of these things are the action on the given function, right? There are definitely, there are other mechanisms of change. I was just dealing with essentially point mutations or small numbers of small mutations by assuming continuity. That certainly, now you could get around that by saying that that's included in the noise model, but it's clear that there's some interesting function there. The example of this is that, firstly, the space of maps from the circle into spaces, as opposed to spaces themselves, that's the basic insight of string theory, right? Things change when you do that. It's been known that loop spaces and free loop spaces have interesting structures on their homology, and maybe, this kind of interaction of things, right? Having two related gene sequences that could go ahead and interact in an interesting way might define something like a product. I have this space, call it S, S cross S into S, right? That isn't a matter I take a curve and I concatenate it with another one. That's a classical thing called the Pontryagin product. Okay? But this is a really, really different thing. And the question is there some way to model it? Will this teach topologists something new about loop spaces? And things like that. Okay, anyway, so all of this was basically my way of saying thank you biologists, thank you universe for creating organisms. All of these things give us interesting topology to think about, and now and in the future. Okay, yeah.
Mike, okay. I apologize if this is slightly off topic. But you're talking about persistent homology and the topological spaces, and in some cases, there's been recent interest in understanding whether the dynamics of the space are occurring only on the edges of the spaces in like a lower dimensional hyperspace, or whether they're filling the interior. And I'm wondering if the topics you're investigating can comment on whether you're getting things that are happening only at the edges, or if they're actually happening, say, in the entire space.
So I'm not absolutely sure that I understand the question, so I'll answer the question that I think you're asking. Okay, and so think about the sphere and a mushroom, okay? And suppose that I have some, so again, all the curves there, it's a sphere, so they all fill in very, in the natural picture, they all fill in pretty easily, right? But I have one that goes around the mushroom twice. Okay, so that would be the kind of thing that you could measure, that you need, that in the course of the deformation, it would be necessary to cover every point of the mushroom 2 times, not necessarily at the same time, but twice the center of any point in that mushroom, right? At some point in time during the deformation will be hit and might be hit a second time. Okay, so that's the kind of information that you could get out of this. So yes, and it's within this bailiwick. It isn't a special case of anything I said. Yeah.
On mushrooms or mushrooms growing next to each other? Well, somewhere separately on spheres. Do you allow?
Okay. Yes. So that's, yeah. That's a different, beautiful, infinite dimensional space, okay? The space of metrics on the two-sphere, or the space of metrics on some n-dimensional manifold. So, right, so, you know, so I was just thinking about the mushrooms as being giving a particular kind of metrics. Then, of course, if I do mushrooms on mushrooms, right, you could imagine, given any tree, then taking, you know, and your tree is embedded in space, I could take a little neighborhood of the tree, and that will define for me some kind of metric sphere. Okay, and so for the problem, for the log L problems, I was saying that nothing was noticing the difference of which point I was on that sphere, right? So that's what topologists do. They might use the metric, but they're not supposed to care about it. Okay, but now, you know, I mean, I'm more enlightened, right? So that was why, that was the very first question that, you know, in my list, this sort of to-do list is, are these kind of things relevant and interesting in how it changes based on the metric? That's sort of this kind of finer kind of things, right? Do you care about whether, and I think that probably, you know, it's sort of asking, do I need a little bit more energy, right, rather than double the energy, right? I mean, I'm saying that you don't have to increase the complexity more than double. Okay, and now why do I pick double or triple? Well, that's because I want to be metric insensitive, because it doesn't matter whether I'm measuring in meters or kilometers if I'm just going to be asking about doubling, right? But if I'm going to be asking, I have to pay $10 for the extra stuff, I'm going to be charged, then maybe I do care about these kind of questions.
Well, I apologize if this is kind of obvious, but I'm trying to follow the general message. So what you're saying is that if we take all these networks and interactions in a hyperdimensional space, they all will tend to look the same.
No. No. I say each network, firstly, there are lots of ones that look the same that you really should distinguish by the higher dimensional things. Don't just measure the pairwise things, which is measured in the thing. Measure about which triples are simultaneously available, which quadruples are. Okay, and then when you do that, you get lots and lots of spaces. So you see, right? The theorem was that the persistent homology is the same as that there aren't any long bars, which means, roughly speaking, that that you don't have to increase complexity nonlinearly in order to solve problems. Okay, but it doesn't solve the problems for you. That's involving to know what the answer is. It says, oh, you speak to the oracle, and under some good conditions, you could understand what the oracle said. Okay, right? I mean, but it doesn't say that, you know, you're going to get the same answer no matter when you consult the oracle, right? I mean, if you ask me, if I'm looking at, say, maps whose image would be the closed interval, any two things can be deformed to each other. I just take my function and I multiply it by t, let t go to 0, and they all become the constant function. I can't do that on the circle because of winding number, right? But it turns out winding number tells me the whole answer in a gradient flow understandable way, okay? And now for other targets, even though it's not gradient flow, there's some kind of, this theorem implies the existence of something like a pseudo-gradient flow, okay? That doesn't. That's maybe that's a maybe. I might regret that interpretation, but I think that it might be an interpretation of what the theorem says. So thank you again.